Hi everyone, my name is Ashley Gutierrez. I'm a junior in CC majoring in biochemistry, and I'm currently a member of the Hunt Lab, where my project is focused on understanding the role of AUA codons in E. coli. So back in 2016, the Hunt Lab published a paper about how codon influence on protein expression in E. coli correlates with mRNA levels. In this paper, they created a model that can characterize a codon's translational efficiency. Here is a visual representation of that model, where each codon is based upon its correlation with protein expression level. Positive values indicate that the codon increases translational efficiency, and we call these the good codons, while negative scores indicate that the codon decreases translational efficiency, and we call these the bad codons. Pointed out here by the arrow is the worst codon for translational efficiency, the AUA codon, which codes for isoleucine, which my project is focused on. So my main question for my project was just what stereochemical factors were causing the AUA to attenuate protein expression so significantly. To test this, we picked a few genes used in the paper already and redesigned their nucleotide sequence with the worst 30 bad codons from the metric from before. That means there's a high level of AUA content created. Then we simply use the same experimental methods, which is overexpressing our gene of interest in E. coli, together with a PMGK plasmid, which overexpresses the tRNA cognate to the rare codon in arginine, so that our result was not overshadowed or biased by any other rare codons, only AUA. We also did a different set of experiments in which instead of using a PMGK plasmid, we used a plasmid called PRL, or codon plus, which overexpresses the tRNA cognate to rare codons in arginine, leucine, and particularly isoleucine. After analyzing our data, we saw the most protein expression attenuation when the tandem AUA codons, which is an AUA-AUA codon or di-AUA codon, were present in the variant. Additionally, by replacing those AUA codons with synonymous AUU codons, the expression improved in both genes. However, this phenomenon seemed to be dependent based on the levels of tRNA available, which could get depleted rather quickly during the overexpression of our genes. As you can see in the PRL gel, expression was only restored in the variants where AUA was present, but no significant change was seen in the ones where the codon was absent, suggesting that the depletion of the tRNA only happened with disproportionate usage of the AUA codon. Lastly, since we saw the highest level of protein expression attenuation with the tandem AUA codons, we looked at 3 additional genes in which their natural gene sequences were about the same length and contained the similar numbers of multiple tandem AUAs like di and tri, but little to none of the mono AUAs. Not surprisingly, the tandems attenuated the expression significantly, but the attenuation seemed to also vary between the 3 genes, suggesting that codon context played a key role in this phenomenon that we're witnessing. Our next steps is to conduct a ribosome profiling, where we expect to see the ribosome stalling while decoding the AUA codon. Here's a summary of our findings, and if you have any questions, please leave them in the comments.